Thank you so much for having me here. Welcome to University of Illinois in Champaign, our quaint little town. Maybe when you guys think of anthropology, the first thing you don't necessarily think about is perhaps genetics. A lot of you guys probably think of the cultural anthropology side. I think that's what we're most well known for. But there is a biological side of it, and we're called biological anthropologists, and that's what I did my doctoral work in. I looked at, and really what biological anthropologists study is human variation and its applications to things like evolution and adaptation. And what I looked at was always an applied sense of that. I specialize in a subfield of biological anthropology, which is forensic anthropology. And I was really interested in looking at human variation from a skeletal perspective. And in my postdoc, I decided to transition into looking at human variation from a genetic perspective and staying within the theme of how you can apply that to forensics. So today I'm going to talk to you guys a little bit about, I've labeled it forensic genomics, but it's really going to be a conversation about the applications. So the talk's about an hour, but please feel free to ask questions throughout. I'll also be staying for lunch afterwards, so if you have any other questions then. So just a brief introduction to genomics as a field. I think really what's happened is that we've had a transition from talking about just explicitly genetics to this term genomics, and I think that this is confusing. It was for me when I started thinking about it. And I think really what the catalyst was historically behind this was thinking about it as sequencing technologies. So you can imagine when we first started understanding DNA and trying to explore the genetic code, the real difficulty, the real limitation was actually the sequencing capabilities. It's extremely expensive, it took a lot of time, and you could only target particular regions because it was so expensive if you wanted to look at a lot. And so really what happened when we had something that all of you I'm sure are familiar with, but next generation sequencing technology really revolutionized the field of genetics, and really is what contributed to us being able to come up with this other field of research which subsumes genetics, which is genomics. And it's this massive, what I have here, massively parallel sequencing, which is a common way to describe next generation sequencing technology. But what it really allows you to do, instead of kind of sequencing one region of the whole genome at once, it allows you to look at lots of sequence fragments, lots of information, lots of genes collectively. And it does it for a relatively low cost. So what you have is you have an increased amount of DNA that you can actually observe and research, and you have it at a decreased cost and a decreased time. And so this has really opened it up to being more feasible. You can imagine when the Human Genome Project started, it cost millions of dollars to sequence one human genome, and it was a massive effort, and it took years. And so now it's changed, and it's constantly changing. It's constantly improving, and that's really what's driving the field, because it's just the limiting factor is really the massive amounts of information when you have to look at the billions of base pairs. So what does this do? What this really allowed and what this opened up was kind of taking a holistic approach to thinking about DNA, and that's really what I think when I think of how I would describe genomics. It's this holistic approach, and it also is really important because it recognizes the need to study more than just the sequence variation and thinking about it as genes themselves. There's more things going on to get the full picture of human variation and expression, and that's things like what shape are the proteins that are actually created from these genes? Epigenetics, which is a kind of brand new, but it's really coming up and popular, and how that's actually influencing the field, and I'll talk a little bit about that. And then the non-coding region, what we thought of was junk DNA, and it's been described that way for so long. We've been realizing, hey, it's not junk DNA. There's lots going on there that can actually inform upon the expression and the regulation of gene function. So just to give you kind of a basic overview of some of the fields, because genomics as a whole is extremely diverse in terms of what you can study. You can spend your whole life studying a tiny little piece of one of these facets. So sequencing technologies, that's a whole field in and of itself. There's people who are devoted to how to streamline and improve sequencing technologies. Just as we have next generation sequencing technologies, there'll be additional things coming out in the future. And for next generation, I just have a little iteration here of what it showed. This is an example of what happens with next generation. Instead of kind of getting an average read of all the genetic material that's in a sample, what it allows you to do is actually get a read of every single one of these little fragments. If you imagine this is all from one person. And what it's really great for, I think, because I study mostly degraded DNA, is you can take a particular location, like there, you see the A for the adenine, the nucleotide, and you can actually say, what is there for every single one of these tiny little genetic fragments of DNA from this person? Are they all A's at that one location? Do we have something else? Do we have a C or a G? And it'll tell you a breakdown of what percent, right? It'll give you quantitative information about, at that site, how many of the fragments conform to the expected nucleotide. Okay? So this is really important because if you have things like contamination in your DNA, if you find things you don't expect, that's one of the places that you can actually, one of the major benefits for me when I think about next generation sequencing technologies. Gene identification and function. You guys have probably heard a lot about this. There's something called genome-wide association studies. It's in the news a lot. And what this is we have an idea of observable characteristics, whether this is disease or something with appearance, or stature, things like that. And we see this a lot in disease studies where you have an idea of the phenotype or that observable characteristic. So if we take an example of people who have type 2 diabetes, right? Those are the patients. And what they do is in a genome-wide association study, they say, okay, we have people who have an observable characteristic. We're interested in seeing what the genetic components are for that. So let's collect individuals who have the phenotype, right? This appearance, this observable characteristic of diabetes. And they say, and then let's compare their genetic variation. Just randomly, let's collect all this genetic information we can. We don't really know what we're looking for. And let's compare those same genetic positions to people who don't have type 2 diabetes. And they try to control for everything else so that really if you see any differences, all you're seeing are differences that are driven or most associated with the difference between the two groups. And in this case, in my example, would be type 2 diabetes. So you get the DNA from your two groups, and then you compare it. And because certain genetic variants are going to be in higher frequencies, and those that are associated with the type 2 diabetes, individuals, researchers are then able to say, okay, well, what do these genetic variants do? What genes are they associated with? And from there, they can actually start to learn the function. So the genome-wide association studies help to identify particular locations on the DNA that are useful. And then from there, other researchers or the same researchers can actually look at the function of those particular genes that those genetic variants are located on. Gene-gene interactions. Most of the observable traits or diseases that we're looking at are not just simple traits, right? They're not just going to be simple Mendelian traits. You're going to have what's called polygenic traits. And so in this case, you have tens to hundreds of genes interacting and influencing the actual outcome or the observed factor that you're looking at. So people study this all the time. Epigenetics and environmental influences on the DNA. A basic definition of epigenetics, which I'm sure you guys will be learning more about this week, is heritable changes in gene expression not attributable to DNA sequence variation. So I'm actually not well versed in epigenetics. It's, like I said, it's pretty new. And I don't think we actually understand a lot of what's going on, but there's things like DNA methylation that are altering the DNA and influencing the actual expression of DNA in ways that aren't necessarily coded in, but are actually heritable. So this is pretty interesting. Then there's people who study the applications. So you can imagine, okay, we have all this genetic information. We've done a genome-wide association study. We know the function of these genes. We know what genes are now associated with something like type 2 diabetes. Can we apply this in the medical field? Some of you guys have probably heard of personalized medicine. This has a lot to do with, we couldn't have done this in the past, but now with the advent of genomics and the ability to look at massive amounts of DNA information at an individual level and at a decent price point, you can do things like personalized genomics at a medical level. We also have things like Ancestry.com, 23andMe. Anybody done that in the class? Yeah. And I have too, and I actually use it in some of my research. So that's more like, for personal recreational use. We also see genomics research in the legal field, in the forensic space. And this is really what I'm going to be talking about today, is more the applications. This is where my work kind of sits, is in between the technology side and its actual application to real world stuff. And there's also, people who are studying, and I work a little bit in this space, the ethical, the legal, and the social context, right? Science isn't in its own, isolated space. Any science that's worth doing has some sort of application or broader impact to society. And that's how you get funding. You argue for your research and how it's going to impact society. Having said that, you need to then actually explore those ethical, legal, and social implications of your work. All right, so let's go a little bit into my work. I'm going to talk about two main research projects I have going on right now. The first one deals with something called forensic DNA phenotyping. And really what we talk about here and what my research goes for is looking at kind of the hurdles of using DNA to predict physical appearance in the forensic context. So here's something, maybe you have or haven't heard of this. This is something new. This is called the Snapshot DNA Phenotyping Service. And this is catered to forensic professionals. And what they say is, hey, you've got some DNA at a crime scene, or you have a body that's skeletonized and you don't have any idea who this person is. You can do more than just look for a match. We can paint a picture. So you can see here they're saying we can predict things like skin, hair, and eye color, whether or not they had freckles. We can estimate their sex using the genetic information. We can also do ancestry, and we can do it from a very small amount of DNA. Okay? So we're going to go into this a little bit more and talk about its implications in the forensic context. And then we're going to lastly talk about humanitarian applications that still are within the medical legal context, and really talking about medical examiners' offices and that are close to the border, and how they go about identifying and repatriating those that have died on the border. It's ever increasing how many are dying, and so how they can improve the quality of those identifications and getting the bodies back to their loved ones. So when we think about forensic DNA phenotyping, the first thing is just to refresh what phenotype means. Phenotype is just observable characteristics. So anything that we can observe, that's a phenotype. Things that are useful in forensics, things like physical appearance, or trying to estimate something like geographic ancestry. And what we know is that phenotype has a large genetic component, okay, but it also is influenced by environment. So it's a combination of your genetics and your environment working together. So what we know is that we can then say that genetic variants, right, are going to explain a portion of the variation of observable characteristics. I have brown hair, I see some blonde hair, there's red hair, right? So a portion of that is going to be explained by your genetics. A portion of it's going to be some kind of environmental factor. The genetic portion, if you think about it, that's if you go to a crime scene, all you're getting is genetic information. You don't have a history. You don't have an idea of what they were exposed to or that environmental component. All you have is the genetic component. So this becomes a DNA estimated body. So imagine you get a DNA sample from a crime scene, or you have remains that are beyond recognition. They're heavily decomposed or skeletonized. You can actually use known genetic variants that are linked to functional prediction of physical appearance, something like pigmentation. And what we need to remember, though, is that when we're doing this, we're only predicting a proportion of it, right? We're not catching that environmental component. So the real question is how much of the phenotypic variation is actually genetic versus environment? Well, what have studies shown so far? Okay. Well, what we see is that here's an example of eye color and hair color. And all that's going on here is these are just the genetic variants, the genotypes that actually differentiate red hair from like an auburn hair from like a red hair. Here's those, I've got them covered, that are associated with blonde hair. These are affiliated with black hair. So these are the sorts of things where they've been able to use something like a genome-wide association study, isolate hair color, and try to find those genes and then look for the functional aspect of them to see if they actually are correlated with something like hair color, and then here's eye color. What you'll notice is that out of all the genetic variants that people have actually found today that are correlated and predictive of pigmentation of hair and eye, we're only capturing about 40 to 60% of the variation is explained. Okay, and then you can see for hair color, it's about 20 to 30%. So what that means is there's other factors playing here that could be environment, or it could just be that they're still genetic and we haven't found them yet, right? We don't know which way it is. My presumption is that we're going to have a lot more genes. It's a lot more complex and polygenic than we realize. I think there's going to be a lot more. That's not my work, but there are, like I said, this is what's cool about genomics. You can have people who are studying the applied side of it, and then you have somebody who's got to go and actually is interested in finding those genes that functionally create hair color and create the differences in hair color. So what's happening? Well, we have these, we can actually, we have some kind of correlation. We know that if you have certain genotypes, you have the odds of having a particular hair color or eye color. So what we've seen is, we've seen research groups start to have papers coming out that talk about, they're packaging it as predictive tools. So you can see here, this came out in 2013. It's called the Hirisplex system. It's called that because iris relating to eye color, and then the H was tagged on for hair, so hair and eye color prediction. From DNA. Here you can see one that says it can predict eye and skin color in diverse populations using 7 SNPs. So I think one of the things that's most important to emphasize, in particularly this HiRiskPlex article, is they say they can provide reliable hair color predictions independent of biogeographic ancestry, so independent of the origin of that individual. So this rubs me a little bit wrong. Let's go over why. Let's do a little test. So to think about why this bothers me, and probably why it'll be a little bit in your mind, is let's think back to about human pigmentation and place. That should be an and, not a percent. We know that pigmentation is geographically structured, which means it's correlated with geography. And this has to do with the fact of how different pigmentations have been mutated, spread throughout the continents, selected for in certain regions. A classic example is something like skin color. We know there's heavier pigmentation of populations who are residing around the equator, and this has a lot to do with the ability of melanin to counteract UV absorption. We want to absorb a certain amount to get vitamin D. However, we don't want to overabsorb. And then you have the opposite happening in populations that are away from the equator. But the problem is that genetics are also geographically structured. Mutations happen somewhere, and do they stay in that area or do they move out? That's all has to do with what those populations that are carrying those, what populations are meeting what populations, who's mating with who. So the question then becomes, are they predicting, since genetics, ancestry, and pigmentation are all related to geography, are these kinds of models predicting pigmentation or place? So I bet without any genetic information, if I said something like, a person that has blonde hair and blue eyes, what kind of ancestry do you think they have? What kind of broad continental ancestry do you think they have? Some kind of Northern European ancestry. Wow, you guys are better than the prediction, right? I mean, it's like, so this is the issue, right? Is that you can have, there's such a strong tie in some regions to pigmentation that we have to make sure that, just sort those out. We have to make sure that you can actually predict pigmentation regardless of the geography. So these are just maps showing, even within Europe, where there's the most pigmentation diversity of hair color, kind of the frequencies of light hair and light eyes. So how did HiRiskPlex set themselves up? Well, they identified and they used 24 SNPs. SNPs are just a version of genetic variants. And they used a statistical prediction model called a multinomial logistic regression. And it was just a suitable statistical model for the kind of data that they were working with. And they developed it, and they developed the actual prediction model on a European sample. And their argument was, hey, there's the most hair color variation in Europe, so we might as well have a sample that's from Europe. And then what they did was they said, well, we got to validate this. And we got to validate how good the predictive model is, so let's validate it on a worldwide population. So what they did was they used something called the Human Genome Diversity Project sample. And this sample is worldwide populations, as they say. However, these individuals in this sample have geographic ancestry to one location. So how they chose their samples, they said, I'm from this village in China, and I've been here for 10 generations. I have, this is what the family has told me, this is our idea. And so these are people who have a strong geographic association with a particular location. So what that means is when you're using that sort of sample to test it on, you're not sorting out pigmentation in place because they're still tied together. So how can we actually test this? One of the things that's really funny is that this is actually a plot from the Walsh et al. Paper, and what they said was, hey, these 24 genetic variants that we're using, they're not just good for predicting hair color, they actually are good for geography. They said, look, if we just plot the genetic variation in a space using that information, you can see that European samples from all different European regions cluster together. You've got the Central South Asian samples clustering together, the East Asian, etc. And the African over here. So look, it's even good for geography, but they never stopped to ask, whether or not it was conflating their results. Were they just predicting ancestry? So how can we separate geography from hair color? That was my question. So the hypothesis then, becomes something like this. Hair color prediction is not independent of geographic origin. Everybody agree, right? We know that it's related, it's correlated. And what that ends up doing is, my hypothesis was that accuracy of the HiRiskPlex model to predict hair is actually inflated, and it's due to that correlation of pigmentation and geographic ancestry. How do you test that then? Well, you got to find a sample of individuals with multiple origin ancestry. So they don't have these simple, I've been in this region forever and ever and ever. So we had to test the Myers-Plex accuracy on individuals with complex ancestries. So we did a study sample, and these are some of my research associates demonstrating how we collected some of the data. We did a pilot study of 105 individuals. I've about 180 to throw into it. I'm in the middle of analyzing it with some students right now to increase our sample size. The individuals ranged from about 18 to 60. A majority of our participants were below 30. We sampled on campus, so we had a breakdown of about 74% undergrads, 11% graduate students, and then 15% non-students that were affiliated with the campus. And we collected a few kinds of data. Qualitative data, we asked them, what's your hair color? We asked them to describe their hair color. Now, I might describe my hair color one way, you guys might describe it a different way. There's a lot of subjectivity in that, right? So we had to also qualify it. So we actually did things called reflectometer hair readings. This is a reflectometer. It's actually used a lot of times for reading skin pigmentation, but you can use it on anything. So to read the pigmentation of anything, the table, the desk, whatever, your hair. I wouldn't put it in your eye because it works by a laser, but so you can see this is an example of how we collected the data for hair color. We also we collected saliva samples, and we ran an estimate for their genetically derived ancestry. And this is provided to us by a % breakdown of continental ancestry. And then we have information we were able to also collect from that same saliva sample, the SNPs that were used, the 24 that were used in the Myers-Plex, so we could actually run the prediction itself. So this is just an example of if you use that reflectometer, what you get. So the reflectometer takes two readings. They take something called the melanin index. Melanin is just going to be what's actually going to color our skin, right? And so the more melanin you have, the more amount of light that's absorbed, and so the higher the reading. So the way that it works is the laser is shot onto the skin or the hair, and then whatever is reflected back, that's what's measured. Okay? And so, but it gives an inverse. It's counterintuitive. So what happens is that higher melanin indices equal darker hair. And erythema is another thing that it measures, and that's because they're typically used for the skin, and so that measures the redness, erythema, right, erythrocytes, red blood cells. So it's measuring the redness, and that would be important, right, if you had variations in something like blood pressure, right, or the person had just gone for a jog and then you read it. For the skin, or for the hair, it's nice too because we do have redness, right, in the hair. That's one of the values you can measure. And so I plotted this, and you can see a spread, right? You can see that what happens is people who actually said I have light brown hair plot separately than individuals who tend to have a darker hue, right? So you can see that what's interesting though is that dark brown and black really don't sort out. Just from these two measures, right? That makes sense. That's fine. It's just two data points, right? So this is how we went through it, just to give you an example of how we arrived at our estimates. So we took a subject, we ran, these are just the SNPs. Remember there were 24, so I'm only showing you a few here. There's 3 options for the genotype. You can have a homozygous or a heterozygous or a homozygous, right? And so they had a 0, 1, or a 2. And then from that, it ran through the nominal logistic model. And it gives you probabilities. It predicts hair color, and it has associated probabilities. If you sum these together, these 4 numbers here, they're going to about equal 1. And that's because the overall probability is going to be 100%. And you can see here that for this individual, based on their genotypes, they're about 85% probability that they're going to have black hair. Okay? And then they also did a lightness and a darkness scale on the Myers-Plex. And you can see that this is going to be quite a dark hue. Okay? So then we would look at the self-reported. And in this case, this subject said I have brown hair. Not dark brown, not black. And so the question is that accurate? We would say yes or no. Okay? And then we would also compare whether they were single or multiple ancestry origins. So we had both in our group. So we did this for all 105. And then we wanted to compare the accuracies. So let's look first at our individuals who have simple ancestries or single ancestries. So you can see here that what we're looking at is 3 different hair colors, okay? And you can see here we've got the % of accuracy of the predictions, so how good the predictions were. Dark brown was 100% of the time, black was about 80% of the time, and light brown was lower. This is almost identical to what they got in Myers-Plex's study. That's great, right? That means that when I'm still using simple ancestries, I'm getting the same thing. What happens when we use complex? We see a major decrease, okay? And these are p-values, so this is just a statistical test for the actual quality of the difference. Is there really a significant difference? And for this one, there definitely is, and for this one, it's approaching significance. Dark brown doesn't have so much of a difference. And there's other logistical reasons that I don't have time to get into today about why I think this is happening. But what you can see that overall, we do have decrease. So it does mean, in this case, that ancestry probably is playing a role in what's being predicted, because when you complicate ancestry, the model no longer functions as well. So it is predicting hair somewhat, but it's also predicting ancestry. And that makes sense because, genetics and variables, they're complex. They can actually, represent multiple things. And because of those correlations between pigmentation and geography, that's what we're seeing here. So we've got to be smart and refine this model to actually be able to take ancestry into account and identify it and its role in this predictive process. So what are the implications? I think what we need to remember is that when we put out papers as scientists in a research space, and we make claims, and we claim that they've been validated, and then we make claims like it's independent of geographic ancestry because we tested it, we have to always contextualize those within the society at large. And I think that because this is a European research group, they really were looking at more the European population diversity. In the US, we have a much more diverse population. Complex ancestries are very common. And what we have to do is we have to then test the reality of the application to our specific population. And what this does is this means that the scientists can't just operate in their own bubble. They have to be informed about the application of their work and where it's actually going to be used. And in this case, the reality is that it's not going to function well on a complex sample. So if we wanted to bring this into the United States right now, the quality of the predictions would be quite low. So I think what it comes down to is that we have the ability, and just because we have the ability doesn't mean that it's ready to be packaged and marketed. And this is something that, as a scientist, sometimes we don't think about this side of it. And so I think that the other thing that's pretty interesting with genetic technologies is that genetics in general, particularly in the forensic context, has been held up as the gold standard. Things like fingerprints, blood spatter, these have all come under fire the last few years. Genetics does not, because it's viewed objectively. You have a sequence. The sequence doesn't change. You can get objective, factual information from that. But at some point, there's an interpretation, whether that's a statistical interpretation, right? A subjective interpretation of what a brown hair color means versus a black hair color. So what I'm talking about here is that because of this idea of the gold standard, we have to be extremely conservative with starting to apply it in ways that stretch beyond the objective information of the genetics. So we have to look at things like research biases.
So has the forensics field, so like looking at it from a legal perspective or a law enforcement perspective, have they actually started to move towards implementation of these types of things?
So the ancestry part we have seen used, and it's really come under fire because of, which I'm going to pull in next. So it has been used in a handful of cases. We actually have laws in many states that don't allow investigations to use genetic physical information at this point. Not all states have it. It's at a state level, but I think like about half of them have it at this point. In Europe, there's a few countries that are allowing it, and that's why a lot of this research is being driven. But I can see this is what I think is really important is what's kind of nice. What happens in the medical field, a lot of times the genomic technologies are there, we do this stuff, and then maybe what happens is we realize, we shouldn't have done this, or it doesn't make any sense. Like, there's these tests where they can test you for like 40,000 diseases or something, and the doctors say, great that we can do this. When do I have time to sit down and interpret these results? I don't. What's the point of this test? So you catch it on what happens is you have these genomic technologies, they get packaged, marketed, people start using them, and then they realize, this is stupid, why are we doing this? What's cool and kind of unfortunate for forensic space, but most of the local law enforcement agencies don't have money. They barely have money to do basic CODIS, STR, the standard genetic tests, much less something like this that is still in the early stages. It's not at every state crime lab yet, which means it would be a unique situation. But what's great about that is as scientists and researchers interested in the social context of this gives us time before it's packaged and kind of shot out there, particularly in the forensic context, whatever repercussions could there be of predicting the wrong skin color or predicting the wrong hair color. Eyewitness, it's kind of like an eyewitness, but like a DNA eyewitness. And I can imagine people thinking, there's no bias. we should convict.
Right.
And that's what's really scary, right? So it's nice that we actually have this lag time before it's been heavily incorporated to give us this time to critique it and think about it in that real world space, you know? So we'll see. I mean, I think as it gets cheaper, you're gonna have to integrate it, but everything has to go through everything's really slow in forensic science because in the actual applied side of it, the research is going crazy because it's such a popular field. But actually getting integrated at a state level crime lab, the protocols and the quality control is so tedious and slow. It takes a lot of time. So there's research biases, and this is good. That's what we're taking the time right now, kind of in this lag time between it being research and applied to explore. And there's also going to be practitioner biases. What is an investigator doing with this information? You know, they get this breakdown of predictive answer. Do they know how to interpret this? Do they know do they know what the caveats are, or the hurdles? Where's that line of communication between the scientists and the practitioners, right? So this leads me to think about some other ways, and we're going to give an example of some of these biases that we probably all have as lay people, but I think investigators would have as well, and that scientists, you know, also have and need to be aware of how this works. So, interpreting, right? That's what I'm thinking about here, is interpreting DNA when we think of it as this kind of quantitative, objective fact. So one of the things that I think is happening a lot is you're getting things like genetic ancestry and physical appearance, and they're operating, our crime and our forensic units in the US operate in a racialized way. When they think about descriptions of individuals, they use race terminology. Any missing persons report or case, even if somebody is dead and they're doing an autopsy, there is an expectation to estimate race from a body, okay, alone, with no other information. So there's a big emphasis on the fact that people have an ability, and you should be able to estimate race based on physical appearance alone. And genetic ancestry has kind of fallen into that because of its relationship, like we just shown, with physical appearance. So an example of this that's happened in the past decade is there was a Louisiana serial killer in the early 2000s. And they got desperate. They couldn't catch a lead. They didn't know who the individual was. And about that time is when genetic ancestry was coming into play. And so the investigation was getting a lot of news, and they said, hey, let's run genetic ancestry on this guy. And so they did an ancestry breakdown, and they said 85% African ancestry, 15% Native American ancestry. And the investigators have a quote saying, when they were explaining to the public, individuals unlikely to appear white. Okay, what does that mean, right? So why I have appear italicized here is because this is exactly where you can see you have ancestry and physical appearance being linked to a race, okay? And maybe you guys are going, so what? Somebody who has about 85% African ancestry, I would think they would, maybe appear some way that doesn't look white. Maybe that's what you're thinking. But where is that threshold for you? 75%, 65%, 50%? It's gonna differ for all of us, okay? This is highly subjective. So we're running from the objective of just a percent breakdown to now subjective. And we've got this relationship here between race and physical appearance. This is a highly embedded, ingrained US societal norm of thinking that you can extrapolate someone's race from physical appearance. And then through genetics, we start to talk about this relationship between ancestry and physical appearance. So there's a natural transition to say, well, hey, let's complete the circle. These all are equivalent. So the question is, how valid is this presumed relationship? Well, let's give you guys an example. So what this is data from myself and my collaborators that we presented last year at a conference. And what you can see along the x-axis, this is 0 to 100%. So this is a proportion of West African ancestry in an individual's genome. So you can see there's individuals all the way from 0 to basically 100%. And what we have along this axis is melanin index. So remember, we're talking about the pigmentation. Higher pigmentation equals a darker hue, okay? And what you can see is there a relationship, right? So as you increase, okay, African ancestry in the genome, overall, you're going to see a general trend of increase in melanin index. Great. The correlation is about 72%. It's highly significant. Okay, so let's just do a little breakdown here. What that would mean for this person right here, each of these little dots represents an individual. So there are hundreds in this sample, I think about 600. This person right here has about 25% West African ancestry, and their melanin index is about 50. So that's how you would interpret this for each individual. But even though there's a correlation here, right, and this may go along with what you would have thought, forensics doesn't operate along trends of statistics. It's a case-by-case basis. So I want you guys to think about this. Okay, so let's take a little line here. And let's say we have somebody who has 72% about West African ancestry. I want you to notice the range of melanin indices that are possible, okay, that just for this sample that have been included. So to give you guys an example of images, here's an individual who has 72% West African ancestry, and her melanin index is quite high at 68. What about a little bit lower? Okay, here you have somebody who's the same 72%, and the melanin index is 45. All right, look here. 72% West African ancestry, melanin index of 20. So these presumed concepts of what race and ancestry mean together and how that can relate to a suspect description are completely invalid on a point-by-point basis. We can look all day and see that there is a trend, but when it comes down to a single case and you're getting an output of 72% West African ancestry, what I'm pointing out here is phenotypically, we cannot be sure. And you can imagine this kind of information coming out, like the articles I showed you, where it says we can predict skin color. What does that really mean, right? What does predicting ancestry do for a case? And this is what we are concerned about when we think about the social, legal, and ethical implications. People have a conceived idea of what 72% European or Asian or African, whatever ancestry would look like. And the reality is that this is not something we can draw a proxy to race. Race is extremely complex. It's a socially, socially constructed idea, okay? And it's tenuously tied to what we have placed on it as physical appearance. And that relationship is tenuously tied to ancestry. So this is something that I think is really interesting and important for people to realize when we talk about genetics, right? There's a high amount of variation, and we can think about it as, this is raw genetic data, but how we put that into the forensic context in terms of the race context in forensics, we just have to be highly conservative. So I think what the take-home message for this part is really that it's exciting to use DNA, right, in these dynamic ways. Before, in the forensic context, we were just taking these markers and trying to match up. If we have a missing persons or an unidentified persons or a suspect, can we match them with a family member to see if that's their DNA? Or you get a suspect that you think it is and you match their sample to the DNA at the scene. That's a very kind of standard way. So these dynamic ways of saying, look, we can do all this other stuff, it's great. But we really need to understand the limitations of that sort of genomic data. And I think that jumping the gun and applying it before we really thought about the implications is extremely important. So we really need to ensure that scientists and practitioners are working together. And this is not just in the forensic context. This is in the medical field, okay, in the legal field, in the public sector of things like 23andMe. It's very important that this relationship is honed and developed between the scientists and the practitioners. And the applicability of the research, right? I mean, if like we said, you can predict hair color all day. How useful is that really in a crime scene if it's a suspect? They can dye their hair, right? So again, it's kind of like the nuance of, oh, we can do it, but how useful is it really for the practitioners? So again, it's the scientists working with the practitioners. What's useful to you? What do you see? What kinds of things could genetics do that would actually, help you instead of presuming we know how that field works, right? So, the responsibility of science to society, and that's in any kind of science, not just genomic science, is the validation and the quality control and ensuring appropriate applications. So, if we think about the responsibility to science and society, we can go above and beyond something like the creation of, missing persons, visible profiles or whatnot. We can actually use it for something like what's happening at the border. So just a quick introduction. This is actually a proportion here, so just kind of ignore these numbers. But what I want you to do is see the overall trend here of sweeping upward in relation to fiscal year. You can see this goes from 1990 to 2012, and what this is showing you is the deaths along the Tucson border, particularly in Pima County, which is, along the Arizona border, particularly in Pima County, which is right in Tucson, Arizona. And what you can see is that there's been about 2,238 deaths in the last 2 decades. However, there's been a massive spike over time. So the question is, what's driving this? What has changed that's driving this? We've seen hyper-militarization of the border. Why are the number of apprehensions and deaths going up still? That ratio. So just to give you a little bit of background if you aren't familiar with what's happening at the border, in the mid-1990s, the federal government decided to move forward with an action basically called prevention through deterrence. And this is prevention of individuals crossing the US-Mexico border through deterrence. And how were they going to deter them? Well, there were several ways. First was they chose to put up a wall along several portions, not the whole region. They chose explicitly their regions of where they were going to place the walls. And they also did segmented, like again, they were being very smart about how they chose where they were going to militarize the border. So you had Border Patrol patrolling particular regions. Now what this did was, this deterred the border crossers to go through particular crossing routes. And the US government knew full well when it was doing this that it was deterring individuals and funneling, which is the funnel effect, funneling migrants from Mexico into some of the most dangerous regions of the desert, okay, to cross and make it. So what this did was, this is what attributed to the massive increase in deaths, because now the only viable routes are those that are most dangerous. And lo and behold, Tucson and their medical examiner's office falls along one of those routes. Now, why are they crossing, and why do we see that escalation? In the mid-1990s, we also see the NAFTA agreement come into play. This has a devastating effect on Mexican farmers. They cannot compete with the prices with that agreement of other countries. They lose their land, or they have really reduced income, and as a result, they have no other option than to try to go somewhere where they can actually make money, and they're moving across the border. So the combination of this prevention through deterrence and NAFTA really have created these dangerous and deadly spaces and corridors.
Can I ask a question? What makes them more dangerous?
So it's because they're longer stretches of desert,
okay,
and there's not as many towns across them.
Okay.
So to get across them, they tend to be, they're also very like mountainous,
okay,
or just highly hyper desert. So it's just a lot longer. So if some of them it may have taken 1 or 2 days from the U.S.-Mexico border, like the point of leaving like a decent town where you have water,
right,
to the next point you can get water. Maybe it was like 24 hours. But for Tucson, it's about 5 to 8 days. And you can imagine trying to carry water for 8 days. And the temperatures, I worked there over the summer, it was about 108 on average. So.
Is the NAFTA?
Mm-hmm.
NAFTA?
Mm-hmm. Yeah. The North American Free Trade Agreement. And if you guys are interested, I have some nice, I should have brought those, but I can try to track down some really nice papers that outline kind of put into context all of this, and that's always nice to have. I just don't have time to go into it much. So what's the logistics of identifying hundreds of thousands of individuals? So you can imagine Tucson is, it's a bustling, city, but it wasn't equipped to have this kind of increase in its numbers. They had one forensic anthropologist on staff at the time when they started having this increase. Now they have funding to have individuals come in and work there over the summer months when there's the most deaths. That's how I fit in. I was one of those that was hired temporarily. So what are some of the hurdles and identifications? So there's standard hurdles always in identifying somebody, right? In any context in the US, you find a missing person, they're John or Jane Doe, how do you identify them? But there's things that are unique to the situation on the border. So if we think about these medical and these examiners and these coroner's offices, this is where all dead human remains from the US side are going to go to the county that the body was found in. That's how it works. So the complex nature of the crisis on the border is what's really driving this issue. You can imagine things like you have a body. You think they're from Mexico. Maybe they have a Mexican voting card. Who is that ME's office supposed to call? Does anybody speak Spanish in that office? How are they going to communicate that they think they have a Mexican national in their remains, right? In their set of remains. This is actually an image from a nice news report that was done on Pima County. There's been lots. Pima County Office of the Medical Examiner has been in the news a lot about this. They're really highly active in trying to let the public be aware of what's happening. Things like the border crosser demographic is also complex. You can imagine if a body comes in and they're a John or Jane Doe, right? There might be things that are unique about them. Maybe their height, maybe their interesting tattoos, maybe their personal effects that they had. Well, the problem is that the border crosser demographic is pretty similar. You have a bunch of people who are crossing the border and dying that are mid-20s or early 20s or 30s, mostly male, and they're all Latino. So they all fit the same demographic. So when you say, I'm looking for a young adult male that died crossing the border, guess what? Everybody is that. There's nothing unique. A lot of them have very few personal effects. The decomposition rate is so fast out there. So you have this rapid degradation of the body. Any identifiable features or photographs or tattoos you lose all those within days out in the desert because the decomposition is so extreme. Sometimes they can get a fingerprint, and that's good if they've been sent across the border before and they came back and maybe died again. If they got caught by Border Patrol, they can use the fingerprint. Even if the skin desiccates and it's super dehydrated, they can rehydrate that fingerprint and roll it and try to get it. But you'll be lucky if you find a hand. Coyotes come, they tear the bodies apart, they pull hands away. It's the desert. It's really hard to find the body.
I'm in Mexico, and I know, but I would think the degradation, why is it? Is it scavengers? Because I would think the low humidity would actually preserve a lot of.
So what you have is, well, what happens is the heat drives the cells to burst, and so you're having that rapid breakdown of the cellular structure. And you can have mummification happen. So I had a lot of cases where you would maybe have a face, but it's so mummified, you can't, there's, the physical features that you would see aren't going to be there. Sometimes you do have it, but the scavengers are really, really heavy. We saw a lot of carnivore damage. A lot of times you would just find a head. So I mean, like, I can't tell you, and that's actually a really interesting question. I'm not sure what's happening, but a lot of the cases would just be a head. So they just articulated the body, and they'll do it rather rapidly. So yeah, I mean, you understand what that space is like. Yeah. This also is really bad for DNA, right? DNA likes cold, wet environments to be preserved, and now you've got hot, dry. So, you're not gonna be able to get the kind of data. Not everybody can you get the traditional genetic markers from to see if you can make an identification. There's another problem with making matches, right? A lot of times if you have a John or Jane Doe, you run their DNA against a family reference missing persons database, right? That's in the US. What happens if all your family is in Mexico, or they're in the US, but they're not gonna come forward for fear of deportation to actually report a missing person and give their DNA? So now you have this DNA. Who do you run it against? So you can see that this is very different than the traditional identification process. There's lots of other hurdles that are going on in this space. So the traditional approach to identifying John and Jane Doe isn't enough. And what's phenomenal about Pima County is that they recognize that regardless of whether or not these individuals are crossing the border illegally, they're humans, and they deserve, even in death, to be respected. And so they're trying to get these individuals and treat them as humans and give them all the benefits that they would if they were US citizens for being identified. So what are some of the logistics of this? Well,
How long do they normally spend trying to identify somebody before saying we have no leads?
Yeah. What's nice is you can do a full report, which you would do on anybody, right? Because you can't presume where they're from or who they are. So we do a standard report on everybody, gets the same kind of full analysis that you can. And then from there, that body is there. You have that data, and if you do the standard search for a match, and if you can't get it, you can't get it. But what's good is that it's not like your data goes away. So if you have a family member come forward later and give their DNA, they do routine runs of the data again to just see if anybody matches. And so you can have, and this does happen years later. They've had a case that was from like 2005. They get a match in 2012 because the family members finally caught word of the fact that, hey, there's actually a place that's doing this, So you can get matches later.
Will they cremate the bodies in the meantime?
Yes. So what's happened, and this changes, and this is very county specific, but for Tucson County, and I can't remember the exact time frame, but it's quite a short time frame. I want to say somewhere between 2 weeks and 2 months, they are required to cremate the bodies or bury them. And this is because they're unidentified and they haven't been identified. And that's because it costs so much money to keep those bodies because they keep them in freezers and in coolers. And you can imagine, that's anywhere, it's going to cost so much money to keep it at that temperature. And so there's so much money that goes into that. So here in Champaign County, we have like a 3-body freezer because we're very small. There, I forget what their capacity was, but they could keep, a good probably like 50 to 100 remains. But that's still not anywhere near. They're not just, they're serving their whole entire county. So the unidentified that are presumably died attempting to cross the border are just a component of it, of who they're identifying.
How many deaths were there in the last couple of years?
They get several hundred each summer. So the summer that I worked there in 2011, I think we had about 140. That's just in that county alone. And you can imagine how big the border is, right? And there's other counties. So this is very difficult from a logistical perspective as well, because these ME's office, these coroner's offices, they don't have a large budget, and there's no federal support for this massive change and increase, right? And so, I mean, anthropologists have gone and talked to some of these other offices, and they've had people actually say, if there's a body that shows up in a river, I'll just kick it back and let it go to the next county. But there's such a sacrifice of being a human that when you make a choice like that to do that to a body, our coroners shouldn't have to make these decisions. But I think it's coupled also with the attitudes towards border crossers. So you're getting an attitude of they're already kind of subhuman or they're illegals, so they're able to separate themselves, and so they're able to kind of mistreat them in that way. But it's also pressure, I think, a fiscal issue, as well. So it's a really complex issue of how they're overworked. I mean, these medical examiner offices are just completely inundated with cases and don't have enough time to do all of them. And it's really commendable what they're trying to do, I think. So one of the things that's been great about Pima County is they've actually they understand that anthropologists could play a major role in helping the identifications. And that, when you have the traditional sense and they have a traditional forensic practitioners that works well in the US, but for this kind of dynamic crisis, it's not working as well. So what we see things like community and institution bridge building. So we have things where Pima County has hired Pima County Office of the Medical Examiner has hired Robin Reinecke, a cultural anthropologist. She's been working there for years. And her main purpose is she actually collects. She's fluent in Spanish. She goes all over the country as well as Mexico and Guatemala to collect missing person information, collect DNA samples, and she has a line in the Pima County Office of the Medical Examiner to take calls every day and fill out missing persons reports so that when they have something like a unique tattoo or whatnot or DNA, they can actually compare that to the family reference database or to these missing persons reports. And the individuals are much more willing to talk to somebody if they speak Spanish fluently. They're not an official, like she's not a law enforcement agent, so that's who handles the cases. So I think they feel much more comfortable. DNA databases, like I said, they've created their own, right? We have databases in the US that are federal databases. Anybody who's missing or a suspect, you can run it across the database to see if you get matches. What Pima County has done is they've actually created a private DNA database for these. So they're just comparing the matches to individuals who do choose to come forward and give their samples. They also work with the Mexican consulates, right? This is a real good way to alleviate the issue of working. It's a multinational crisis, right? So they actually come forward and every time they get a case, let's say the case has a body comes in and associated with this person is a Mexican voting card. They'll go to the Mexican consulate, they meet with them quite regularly, and they come forward with, our investigators say, look, this case, X, Y, Z, whatever, it has a Mexican voting card associated with it. This to us means that they're likely Mexican. Will you pay for the DNA to actually be run? And they will decide whether or not this case should be run by the DNA. They'll do the same thing if they find quetzales, which are the Guatemalan currency, things like that. They'll say, we think this is a Guatemalan national. They'll go to the Guatemalan consulate and ask the Guatemalan consulate to pay for the DNA. So of that person, of that individual to be run. So the governments are working together to make sure that these individuals we're able to afford all the kinds of data that can help identify them. And then they're actually very supportive at this IMI's office of research. They really realize that because they need atraditional ways to make identifications. So they allow researchers like myself to come in and work there and work with the data. So what this ends up accumulating into is a groundbreaking approach to a really complex issue. And this is something that's unique to Pima County. And they're just fortunate enough that they've together been able to get grants and the support, the financial support to actually run this sort of model. But not every office along county coroners or medical examiner's office along the border is as fortunate. So you see this phenomenal approach they've taken, and yet, what do we see? We see an annual increase in the number of unidentified border crossers. And we also know that identification rates are down, and I put approximate here because I can't remember exactly what he said last time, but the lead forensic anthropologist said, yeah, over the last few years, we're actually declining in identification rates. So they have this state-of-the-art approach to identification they've created, that they've honed particularly to their problem. Why isn't it working as well as it was, or as well as it should? Well, the genetic research itself can provide some sort of insight to this. So now we're going to go back to genetic ancestry, and that's what we're going to use here to think about this problem. So these are all the cases across the years that have some sort of DNA associated with them. there are more. These are just the ones that I have access to. And older cases, it's really hard because of what you guys were very keen to ask, how long they keep bodies around. A lot of this is kind of before we knew DNA could work, or the quality of the DNA, even if they have skeletal material, they may still around. It's not, maybe they can't get viable DNA out of it. So this is kind of what the setup looks like right now for the number of cases with DNA. So just pure count along this side. So I have about 350 cases from this IME's office in a range of about 40 years. And what I did was I was able to take that genetic data and I was able to estimate ancestry. And the reason I only looked at Native American and European ancestry is because most genetic studies that assess ancestry in Mexicans, modern day Mexicans, what they've done is they've been able to say it's largely a component between Mexican and European ancestry. There is a low consistent components of African ancestry, but its majority is driven by European and Native American ancestry. Higher Native American ancestry is typically associated with indigenous populations, and something like a mix or a high European component is what they call mestizos. So that's going to come up later, which is why I want to introduce it. And I use these CODIS STRs, which is just the standard forensic genetic identification data. So the question I wanted to ask first is, who's dying? And so what I found was that there's a trend of Native American ancestry increasing over years of the case. So back in the 70s, and I kind of grouped them here just so you could visually see the trend, but from these are like 70s to mid 80s all the way up. And what you can see is there's a slight increase in the amount of Native American ancestry over time. So I wanted to test to see if there's actually a correlation between year of the case and the amount of ancestry. And what I found was, this is the line that was fit to it. You can see there's lots of variation, but overall there is actually a highly significant, statistically significant correlation. So this is a valid correlation. It's positive. So that means as the year of the case increases over time, the amount of Mexican ancestry increases. So what does that mean? Who does that really represent? Well, what we know from genetic studies is that people have done nice surveys. And there's actually been a great, I think it was a Science or Nature article, I think Nature, that just came out talking about the genetic diversity in Mexico and how much there is. But when you look at it from an ancestry perspective, Ruby Castellanos, and I think in 2009, what they essentially showed was that the amount of Native American ancestry in mestizos and indigenous populations, actually it increases as you move from north to south. So you find higher amounts of Native American ancestry in the southern regions. And you actually have the opposite happening with European ancestry. They find individuals with higher amounts of European ancestry in the central and the north regions. So what does this mean? Well, if we think about what it means to have a larger amounts of Native American ancestry, that usually means that, like I said, they're more associated with an indigenous cultural identity. And where do those that have an indigenous identity, some of the largest populations in Mexico today, they're concentrated in the southern regions, okay? So what else is happening in the southern region, okay? So if we think about this from that perspective, you can almost think about this as the mestizo populations being most prominent in the central and the north regions, and the indigenous populations being most prominent in the south. When we look at something like this confirms it. And if we associate it with something like poverty, remember how I was talking about the farmers being driven up north to find work because they've been driven out of it, you can see the lowest amounts of income, annual income, are affiliated with some of those southern indigenous areas. So what we find is that over time, so we go back to that trend of that correlation, over time we're seeing an increase, and this has actually been shown through cultural anthropology work as well. Indigenous populations are starting to move up and migrate into the US much more so than in past years where it was mostly mestizos. And the issue with this is that the indigeneity and the poverty we've kind of made an argument for, but the risk of dying, right? Because this isn't a question of who's crossing. This is a question of who actually makes it and who doesn't. So why are these new kind of indigenous populations maybe at higher risk? Well, social scientists have made an argument that they're at a greater risk of death because they don't have a history and the relationships of crossing like some of these other areas do. So you can imagine that's very dangerous. If you can find a good guide to get you across, or you know a safer trail, or a good way, or you know it actually takes 8 days instead of 3 days like they try to tell you it takes, you know? So, or somebody's gonna try to, you know, take your money and leave you out there, which happens all the time. Individuals who have lived and learned, they can actually pass on that knowledge. But the southern regions, as well as things like Guatemala, they're very new to crossing in large amounts. So they don't have that information, that life-saving information, all right, to cross. And so that's why they have an increased risk of death. They also have to make it all the way through Mexico first, right? So they're crossing longer as well into unknown territories. So the question then is, all right, now we know who's dying, right? We see this trend over time with increased amount of Native American ancestry. Who's actually being identified? Right? That's the next question. Who do you guys think would be identified based on the other trends I told you? We'd probably see an increase over time, right? We'd see an increase of Native American over time with who's identified. And that's a logical expectation. We actually see the opposite happen. So here again is breaking it down. You can see the identified cases have a much smaller range, but we're actually seeing a decrease in Native American ancestry. And because I only tested European and Native American ancestry, that means you actually have an increase in European ancestry. So it's positively correlated with European ancestry and negatively correlated with Native American ancestry. Again, it's not as significant. It's only p.10, which is okay statistically, and then it's negative, 17%. So how is this? This is really confusing to me, because I was thinking, okay, all cases that come through, anybody who dies, right, regardless of where they're from, nobody knows where they're from, they're all getting the maximum amount of data put into the database, right? It's not like they're differentiating, oh, this person must be from the North region or be a mestizo. Let's go ahead and emphasize their case, in terms of getting DNA. That doesn't happen. Nobody knows that. So, why is there a discrepancy? This is one of the questions that I think is really important. And what we find is that there's really the question to think about it is, why is there identification bias towards mestizos? And one of the things I think that's coming through is that it's not on the side of the Pima County. They're doing everything right. They're collecting that data. But you can't make an identification unless some kind of family members come forward. And so I think what's happening is it's the access to the information. The family members are in sometimes they're in very small communities, right? Southern Mexico is much further away than the rest of Mexico. You can imagine these people who are going out, these anthropologists, these NGOs who are going to collect data. It's much easier to go to a decent sized city or town. The infrastructure, the road access to some of these smaller communities, the sending regions, they may never get information that, hey, we can actually help identify your loved one. The relationship between indigenous populations and the government, if you have any idea about Mexican history with the indigenous populations, is not good. They may not be as willing to go to the government to ask for this assistance. And the history of migration in the U.S. context, again, they're not getting word of the fact that there are these social networks that can then put you in contact with the right people to create missing persons reports and maybe look for your missing person. There's also embedded attitudes in the Mexican consulate. Most of them are in the U.S., but they are in the U.S. and they're obviously representing the consulate. But there have been, negative attitudes sometimes towards the poor indigenous populations that are crossing, saying, "Oh, what are they doing?" It's all very informal, but there have been some negative social bias issues. This is relevant as well. So these are the next identification hurdles. You can see Pima County really stepped up and modified their traditional identification protocols when they realized, okay, we're getting a massive amount of undocumented border crossers, but now we're seeing a shift in the identification rates going down. So, if we've got hundreds of cases that are remaining unidentified at Pima County alone, and we still have an increase, an ongoing increase, is increasing every year, guys, and a decreased rate of identification, how are we going to fix this? I think one thing is that we're at a plateau effect. I was talking to the lead forensic anthropologist about this. They modified their cases. They saw a massive increase in the amount of identification rates. But what's happening is the ratio of unidentified to identified is going down every year because the amount of cases coming in is going up every year. But their rate of identification is plateauing. So it makes it look like it's a decrease in the rate. And so I think what's happening is they have to step it up and change their protocols to actually allow this to increase the identifications again. So they've got to change. They've got to be willing to change their approach to identification as the demographic of the migrant shifts, I think. And how can you use this, right? So this is a very atraditional way to think about genomics. This is kind of deeply embedded in this complex web of a crisis. And I think what we can think about this as when we think about what we just saw with these trends is that we're using genomics as evidence for change. When we can show things like an identification bias at an ancestry level, this reflects the need for more community level work. So when we have NGOs like the Colibri Center and the Missing Migrant Project, who are largely run by things like cultural anthropologists, and they work with these medical examiners' offices, this allows them to make a case for we need more individuals who work actually directly with the communities that are not directly affiliated with law enforcement or the government, because there's a negative association, there's a fear factor with that. And I think the other thing to think about is that this goes beyond just something like the border crosser. I mean, if people in the U.S., some people are really also not going to be willing to talk to law enforcement. And in that case, if there's a fear or a negative association, it really does help to have people who are more on the social science side, I think, of identification. So building that community relationship, I think, is where we're going to see that need. We need to have more people going out to the communities, letting them know, explaining to them what's happening with that DNA if they give it, how that can actually improve the identifications and potentially increase the odds of their loved one who has died getting identified. So I think what happens is it justifies collaborative work. Forensics as a whole has been highly scientifically oriented. And I think what this allows us to do is say, look, there's spaces where we could see modifications and protocols that could allow for that relationship between these kind of non-traditional forensic institutions, something like this that works within a medical examiner's office, but it's actually making a difference. And so I think we can actually use this information here to help fund these kinds of projects, not only for the border, but within the US context, I think. There's been an overemphasis on the factual data, and I think that sometimes that relationship and the descriptions that you can get from the individuals themselves who are filing these reports or discussing histories is really useful. So I think when we talk about this as a whole, there's a way to think about traditional genomics, and that in the forensic context, that's really thinking about it as DNA as identification component. You can identify, you can make a one-to-one match. And the problem is, in this case, it's really constrained by a lot of other factors. And so you can't always make that one-to-one match. So you have to create some kind of dynamic genomics. And I think that's where you really see that collaboration between the science and the social sciences to solve problems in a way of thinking outside the box of how to interpret and apply genomic information. And I think what happens when I teach this, and what I've noticed with my forensic science classes, is that it's really exciting, and it can actually be attractive to students in many ways to think about the applied side. What it does is it really provides kind of relevancy or context for kind of abstract context or abstract concepts. When you're talking about nucleotide variation or random probability matches for forensics things, it's kind of like, it's kind of esoteric to the student. And when you can actually bring it into the context and talk about it in terms of like what the risk is of using this or what the benefit is of using this, it's something that they can actually make sense to and emotionally connect with. And I've found that really has drawn students in. I have undergraduates from freshmen to seniors that I've worked with that are really interested in the applied side of it. So I always try to include it in when I talk about genetics. And the other thing that's really cool about the application side is that it's interdisciplinary in nature. As soon as you go to an applied side of anything, you're stepping into another field. And I think this is what has been so cool, is that it's so attractive to many other students. I've had students that want to be cops that work with me on research. I've had people who want to be medical doctors. I've had people who want to be professors in genetics. And it's because it's something that can be simultaneously attractive to many people. If they're attracted to the hard science side of it, there's that component. And if they're attracted to the kind of applied or social side of it, they can relate, they can identify and be excited about something in it. So I really like that part of it. There's also a lot of really good public resources. There's a lot of good blogs and magazines that talk about forensic genomics, forensic genetics, and so it makes it something that's really easy to bring into the teaching experience for me at least, that I've had. And I think it's a way to talk about the broader context of science. And I think this is probably something that as scientists, because we're so focused, we sometimes forget. And I think that as a science for society, it's really important that we always think about the broader context and challenge our students, I think, to think about the broader contexts. So I just want to say thank you very much to you guys for sitting here listening and for your questions. And then here's my email, and then these are just some individuals I wanted to give a shout out to. So thank you very much.